Hi, I'm Yun, the PhD student of the Australian National Insect Collection of CSIRO and the Australian National University. In this talk, I will talk about my PhD project on systematics and evolution of Australian Cycad-associated weevils and our current progress. Cycads are tropical and subtropical gymnosperms with palm-like leaves. They diversified and are widely distributed during the Mesozoic. With nearly 90 species have been described, Australia is one of the diversity centers of cycads in the world. In Australia, during the colony period of cycads, weevil adults will colonize on the male cones, feeding on the pollens and laying eggs in tissues. In the case of Macrozomia, male cones and female cones have similar thermogenesis and volatile emissions. At sunset, when the volatile emissions reach its highest concentration, the weevils will be pushed out and they will be back when the concentration of volatile emissions is low during the daytime. Due to similar appearance and the physiological period of male cones and the female cones, weevils dust with pollens may reach the female cones by mistake and finish the pollination. Cycads are economically important plants. Some of them are usually used in horticulture, and they also have conservation values because many species are listed in the IUCN Red List and threatened by illegal poaching and habitat destruction. Pollination is an important stage for plant reproduction, and therefore we would like to study on their weevil pollinators. So let's go through the current knowledge of Australian cycad weevils fauna. The most important group is Chinese group. It has two genera, Chinese and Molytinae, pollinating three Australian endemic cycad genera. In addition to these two genera, it also has two genera, Straton and the Damiutius, which are trunk borea of cycads, and Paratronies associated with grass trees, and Howeltronies with host plants unknown. Another weevil group pollinating cycads is an undescribed genus and species related to Titioides group in Asia. In our project, we have three major parts. First of all, we want to deal with the taxonomy of Australian cycad-associated weevils and test if the morphological identification is consistent with molecular species delimitation. Second, we want to determine the host specificity based on our taxonomic information and determine their distribution patterns. Finally, we want to know the morphology of Australian cycad weevils and their own systematic placement. Then we want to investigate on the evolution of cycad association in Australian weevils and determine if the divergence type of these weevils consistent with that of Australian cycads. So this is our project diagram. Now we will show you what we have done so far and those ongoing research. For taxonomic section of Australian cycad weevils, after our morphological examination, we have confirmed at least 9 distinct new species for Chinese group and 1 new species for Australian Titioides group. For molecular species delimitation section, we are dealing with the analysis of mitochondrial genomic sequencing data, and for species delimitation, we will use some standard tools like automatic barcode gap discovery. We have completed 2 publications from this chapter. The first one is the taxonomic review of Australian trunk-boring cycad weevils of Damiutius and Silverton. This is a comprehensive taxonomic review of morphology, including adults, larvae, and pupa. These 2 weevil genera can attack non-Australian cycads as well. Among these foreign cycads, we found they have preference to attack African cycads of Enceladus. Based on the cycad phylogeny, we present this preference is rooted in the phylogenetic affinity between African cycads of Enceladus and Australian cycads of Macrozomia and Lepidozomia. In our newly accepted paper on Paratronies associated with grass trees, we also did a comprehensive taxonomic review, and we found 2 distinct species in this genus. Both species show distinct strong distribution in Eastern Australia and Western Australia. Both species are symmetric in distribution and share the same host plants of grass trees. The distinct morphological difference show that ecological character displacement may occur for separation of those species. These two species may utilize different parts of the grass trees in the field. For our second chapter of natural history of Australian cycad weevils, based on our morphological identification, we have confirmed there are only diminutions, Molytinae, and the insignificant species group of Chinese are host-specific. And we also know the distribution pattern of Australian cycad weevils based on our current morphological results. The further molecular species delimitation will provide us more information on their host specificity and distribution pattern. Together with field observations and captive rearing in the lab, we also get some information on the habits of Australian cycad weevils. We also have complete 2 papers from this chapter. The first paper published on Entomological Science is about the biology of Molytinae personalis, the only known and specific pollinator of Bovania cycads. Together with field observations in North Queensland in 2019 and our captive rearing in the lab, we are able to dispute previous statements of ecology of Molytinae personalis in Wilson's master thesis in 2004. This is also our newly accepted paper on bionomics of Australian cycad weevils. This paper is largely based on the recent field trip in North New South Wales in March this year. In this paper, we provide the first observation on ecology of diminutions, meldoides, and report the new host plant record for Ceraton internators. So for our final parts about systematics and evolution of Australian cycad-associated weevils, most of the works are still ongoing, but we will show you the current progress of our research. First of all, we have completed the morphological characteristics of the Chinese group and its related weevil groups such as Osorine and Achilles. We select 131 morphological characters, including adult, larva, and pupa characters. The resulting topology reveals that the Chinese group is a non-monophyletic group because Osorine and Achilles are included in the Chinese group. But the interesting is the close relationship between Chinese insignificant species group and the Chinese lateroid species group is also supported by the molecular analysis of Australian weevils based on few molecular markers proposed by Gunter et al. In 2015. So based on this topology, we can also see the cycad association evolve once in the Chinese group, and then the cycad pollination of Molytinae and the Chinese evolve from the general cycad boring biology of diminutions and the Ceraton. As you can see, our morphological phylogenetic work is just a preliminary study with limited number of the task sampling. So our further plan is to reconstruct a strong phylogenetic framework based on genomic data. We have finished the DNA extraction in the whole genome shotgun sequencing of all available specimens of the Chinese group, Titioides group, and the Molytinae genera and tribes, aiming to build a phylogenetic tree with comprehensive task sampling. And we can discuss more about their systematics and the evolution of cycad association. Now we are dealing with the mitogenomic analysis, so our study is still ongoing. I would like to thank my supervisors from RSP at ANU and Anik of CSIRO, and thank you for your listening.